Hi everyone. Thanks for joining us today. It's 4 o'clock here in the UK in the afternoon. It's pretty early for Freddie and in San Francisco, I think you are.
Seattle, yeah.
Seattle, sorry. On the West Coast. And thanks for joining us. It's going to be another session here. And it will be recorded, and this webinar. Essentially, what's going to happen is that there's going to be two presentations today, but the idea is that there will be a session at the end for answering any questions. But please do feel free to use the chat feature and write any questions that you have, and we'll take them at the end. If you just move on to the next slide, I'll just introduce our two speakers today. We have Freddie Chen, who's a principal PBPK consultant. He received his PhD in pharmaceutics from the University of Washington in Seattle. And he has extensive experience in consultancy projects. So he leads a lot of those projects at PBPK consultancy. Jian is associate principal scientist in Certara Simcyp division in Sheffield. She received her PhD in biopharmaceutical sciences from Illinois at Chicago. She joined in 2017 and has worked on lots of areas within the simulator, including compound files, pediatric, pregnancy, biologics, DDI modules. So she's extensively experienced in lots of different areas of the simulator. And today what they're going to talk about is relating to a publication that Freddie was the primary author for, based on IL-6 and also looking at drug-biologic interactions. And that links really nicely to some of the work that's ongoing at the moment for version 23, which Jan is leading. So she's going to give you an update on that work as well. So let's pass on to Freddie, and you're going to give us the first part of the presentation. Thank you.
So yeah, it's Freddie here. I'm going to be the first presenter of today's presentation, and this presentation is divided into two parts. In part one, I'll be summarizing the current in vitro and vivo evidence of cytokine-mediated CYP suppression, and we'll talk about different cytokines and how they relate, how they compare to IL-6 in terms of their suppressive effect. And this part is really based on the paper that we published last year. It's a collaborative work between me, Hannah, and Kate. Although I'm kind of speaking for Kate here today, to present. She's also here, and you'll see her in the Q&A session. She's really the expert and contributed a lot to this work. And so the goal of part one is really to set a stage up for part two, where Xian will give you a real case study. And so we're really saving, I guess, the interesting part for later. Okay. So I'll begin the presentation with a little bit introduction. So why do we care about biologic and drug interactions? Biologic drugs have been used or been proposed to be used with small molecule drugs for the treatment of different disease areas, in immunologic diseases and oncologic diseases such as rheumatoid arthritis or gastrointestinal colorectal cancers. And an increase or decrease in cytokine levels has been shown to suppress or elevate CYP enzymes. So this PK-driven drug-biologic interactions may arise after co-administration of small molecules and biologics. Therefore, FDA actually requires a language in the therapeutic protein label to indicate whether there is potential for this kind of drug-drug interactions. And despite a clear in vitro and vivo links between IL-6 levels and CYP suppression, in particular CYP3A4, the changes in IL-6 levels in vivo rarely lead to significant drug interactions. And there are PBPK modeling examples in literature showing that PBPK modeling has been used successfully to predict the impact of raised IL-6 on CYP activities. However, beyond IL-6, evidence to support direct effect of other cytokines on CYP activity remains inconclusive. And I'll come back and elaborate on some of these points in later slides. Okay. So showing here is the outline of part 1. Basically, I'll be reviewing in vitro evidence first, followed by in vivo evidence. But in each part, I'll talk about IL-6 first, and then we'll look at other cytokines and how they compare to IL-6. At the end of part 1, I'll highlight some of the examples of PBPK modeling. Okay. So let's dive right into the in vitro evidence, and let's look at IL-6. The majority of the available in vitro data focus on IL-6, and of the cytokines tested in vitro, IL-6 generally causes the greatest CYP suppression, with 3 or 4 being the most sensitive isoform. And most studies usually measure the effect of cytokines at a single concentration that is far in excess of those seen in diseases or after immune modulator administration. For example, IL-6 concentrations of 0.5 to 200 nanogram per mil have been used in vitro, whereas mean values in patients with rheumatoid arthritis or post-surgery patients are falling between around 50 to 30 picogram per mil. And there's also data reported for cancer patients. You can see the number is even lower. But I do want to highlight 2 studies, the Dickman et al. And Evers et al. These are the 2 studies that actually use physiologically relevant concentrations over the range of 0.5 to 10,000 picogram per mil. And this allows us to calculate EMIN and EC50 values, and it's shown on the top right table there. And these values are very important in terms of PBPK modeling because they're the required parameters in order to model this kind of suppressive effect dynamically. And bottom right figure is to highlight the variability in IC50 values between donors. And there's also between laboratory variability discussed in this Evers paper as well. But despite these high variabilities, the reported CYP3A4 activity, EMIN and EC50 values in Evers are consistent with those reported in Dickman. So we do have a level of confidence there. For IL-2, there really has been only 2 in vitro studies. It's kind of summarized here. Incubating human hepatocytes with IL-2 led to no effect on CYP1A2, 2C9, or 3 or 4 mRNA or activity. There is a very modest suppression for 2B6 and 2C19 activity, but no effect on their mRNA. The results for 2D6 is a little bit conflicting. We see an increase in mRNA, but decrease in activity. And the table in the middle is to look at CYP activity and mRNA suppression by IL-2 in comparison to IL-6 in the same in vitro study. And I just want to highlight this last column here as represented by the ratios of % decrease relative to IL-6. So it's really showing you that IL-2 has less of a suppressive action when compared with IL-6. And I'll keep showing the same table for the following cytokines as well. And in one of the study, Simcyp et al. Reported that no suppression of 3 activity by IL-2 in hepatocyte cultures, but they did see a concentration-dependent 50% to 70% suppression in hepatocyte Kupffer cell co-cultures. So this kind of tells us that IL-2 acts via an indirect mechanism, probably through stimulating Kupffer cells to produce other cytokines such as IL-6 to cause suppression. Okay. So for TNF, so for interferon gamma, I'm showing the same table here, but I just want to highlight this last column here. Interferon gamma generally has a similar or reduced suppressive action when compared with IL-6, with the exception of 1A2, where interferon gamma could be a more potent suppressor. There are no reported EMIN or EC50 values describing the interferon gamma suppression of CYPs, and majority of in vitro data have been measured at a single interferon gamma concentration. However, for CYP1A2, we do see concentration-dependent suppression of CYP1A2 activity. This allows us to calculate the EMIN and EC50 values for interferon gamma. But when you compare these values to those for IL-6, it's suggesting that probably interferon gamma is not as potent as a CYP1A2 suppressor as IL-6. And it should be noted that these EC50 values are far in excess when compared to physiologic conditions in disease patients. So the next cytokine we're talking about is TNF-alpha. Again, just want to highlight that reduction in enzyme activities was significant in all CYP isoforms tested and generally are comparable to reduction caused by IL-6, except for CYP1A2, where a greater reduction was observed after TNF-alpha treatment compared to IL-6 treatment. And Klein et al. Actually characterized dose response curves for CYP1A2, 2B6, 2C9, and 34 by measuring mRNA levels in a range of TNF-alpha concentrations. So this allows us, using data from this study, the EMIN and EC50 values of TNF-alpha for these CYPs were determined. And if you look at these numbers, it's suggesting weaker suppression for CYPs compared with IL-6, except for CYP1A2. But despite a stronger effect in CYP1A2 suppression compared to IL-6, it should be noted that these EC50 values were in excess of concentration compared to what's observed in patients with immunologic diseases. For other cytokines, in vitro data for other cytokines are very limited. Currently, there are no in vitro data available in the public domain regarding CYP suppression by IL-8, IL-17, and vascular endothelial growth factor, the VEGF. And there's one sentence mentioned in one of the papers to look at that in vitro studies in human hepatocytes reveal no effect on IL-10 on CYP enzymes even at 5,000 picogram per mil. However, data were not shown in the paper, and it's mentioned as internal data by Amgen. Okay. So far we've looked at single cytokines. What about combined cytokine? Although multiple cytokines are raised simultaneously in disease states or after dosing certain cytokine modulator in vivo, but the majority of in vitro studies have assessed CYP suppression caused by one cytokine rather than the effect of a combination of cytokines as found in vivo. So I want to highlight two papers here, kind of look at the combined effect. On the left here, we have Dickman et al., where they use cryopreserved and fresh human hepatocytes to determine the suppression of several CYPs by IL-1 beta alone or in combination with IL-6 over a range of physiologically relevant concentrations. And what they found is incubating, IL-1 beta with IL-6 together did not really increase the CYP suppression caused by IL-6 alone for CYP1A2 and 2C9. And they even saw a little bit reduced suppressive effect on 2B6. And they saw a small but additive downregulation on CYP3A4 mRNA activity compared to IL-6 alone. And on the right-hand side, in Xi's study, they studied the suppression of CYPs in hepatocyte when incubated with a cocktail of cytokines. and the cocktail contains IL-2, IL-6, IL-10, interferon gamma, and TNF-alpha. They looked at 3 different concentrations. There's low, mid, and high concentrations. And these concentrations were determined using what's observed after blinatumomab dosing to lymphoma patients. And what they saw is similar CYP suppression was observed with the mid and high concentration cocktail, indicating that the suppression is probably already maxed by the mid-strength cytokine levels. And there's very limited suppression observed in most donors for 2C19 and 2D6 activity. And even and there's a greater than 50% suppression of 1A2 and 2C9 and 3A4 activity observed at the highest cytokine levels. So even though they see these suppression and the level of these CYP suppression observed was actually within the range of values reported by other in vitro studies using IL-6, TNF-alpha, or interferon gamma alone. So taken together, this really suggests that suppression by combinations of cytokines probably not likely to increase the extent of CYP suppression compared with incubation with IL-6 alone. Okay, so moving on, we're going to look at in vivo evidence. Changes in circulating cytokines have been linked to alterations in drug metabolism in vivo, and the correlations between raised IL-6 concentrations and CYP activity have been reviewed extensively previously, so we're not actually going to detail here. I'm just highlighting some of the examples for IL-6 here. And raised IL-6 concentrations were correlated with increased CYP1A2, 2C19, and 3A4 activity in patients from different disease areas. It's also associated with increased 2E1 activity and probably does not really have an effect on CYP2D6. And in the green box here shows some of the therapeutic protein drug interaction examples. And there are 2 mAb I'm mentioning here. One's a sarlimab, the other one is a tocilizumab. These are mAbs that blocks IL-6 from binding to its receptor, and therefore they can remove that suppressive effect of raised IL-6. And administering of these mAbs to rheumatoid arthritis patients, we see a decreased exposure for CYP3A4, 2C19, and 2C9 substrates. And there's a slight increase in AUC for CYP1A2 substrate, but again, no effect on 2D6. And another example, COVID-19 patients who received tocilizumab greater than 12 hours prior to lopinavir, which is also a 3A4 substrate, see a significant lower lopinavir exposure. Okay. For IL-2, again in a green box, showing you some of the example of direct dosing of IL-2 or IL-2 antagonist. For IL-2, CYP1A2, 2C, 2E1, and 3A protein expression was reduced in hepatic microsomes isolated from patients undergoing liver surgery who received high doses of IL-2 prior to the surgery. And in a second example, similarly, adenovirus concentration increased after IL-2 administration in HIV patients. However, I do want to mention that significant increases in IL-6 concentration were also observed over IL-2 infusion. And these IL-6 levels are similar to those observed in rheumatoid arthritis patients where IL-6 receptor antagonists were given and led to a 2-fold decrease in CYP3A4 substrate exposure. Therefore, the similar level of interaction between IL-2 and adenovirus suggests that IL-2 probably does not cause an increased suppression of CYP3A4 compared with that caused by IL-6 alone. And another example, basiliximab and daclizumab are mAbs that can act as IL-2 receptor alpha antagonists. So after administering these IL-2 receptor alpha antagonists to renal transplant patients, we see increased levels of tacrolimus and cyclosporine concentrations. And the theory behind this is that the reduction in tacrolimus and cyclosporin clearance is thought to be due to basiliximab blocking the binding of circulating IL-2 to IL-2 receptor alpha on T cells. And instead, IL-2 binds to the IL-2 receptor on hepatic intestinal cells, leading to suppression of CYP3A4. And the last example here, contrarily, administering dexilizumab to multiple sclerosis patients had no effect on the exposure of several CYP substrates. However, I do want to mention a caveat here for the last two studies. The IL-6 concentrations were not monitored in these two studies. And there are a few reports linking IL-8 to CYP suppression in vivo, and no data after direct dosing of IL-8 or IL-8 antagonists. So some of the examples we're looking at here are based on correlation. In patients with psoriasis, IL-8 concentrations were significantly increased. However, no correlation was found between raised IL-8 levels and venlafaxine, which is a 2D6 and PGP substrate. In contrast, raised IL-8 levels in patients with ovarian cancer was significantly associated with increased 2E1 activity and reduced 3A4 activity. However, the changes in enzyme activity were also significantly associated with IL-6 and TNF-alpha levels in these patients. And in fact, the IL-6 concentration and the extent of reduction in the 3A activity in these patients were comparable to those in rheumatoid arthritis patients. So the role of IL-8 is inconclusive here. There's a double-blind crossover study where 8 microgram per kg of IL-10 were administered to healthy volunteers. And the study showed that administration of IL-10 did not really change CYP1A2, 2C9, and 2D6 activities. And the CYP3A activities was reduced by only 12%. Also note that this IL-10 dose level is likely to lead to much higher concentrations compared to those observed in patients with immune disorders. So IL-10 CYP suppression in patients is unlikely. Okay. There are a few reports linking interferon gamma to CYP suppression in vivo. There are no data following direct dosing of interferon gamma or interferon gamma antagonists, unfortunately. Some examples are highlighted here. In healthy subjects suffering the acute viral respiratory infection, we see interferon gamma, interferon alpha concentrations were significantly increased, and antipurine clearance was significantly decreased. And similarly, when HIV patients were treated with zidovudin, the interferon alpha and interferon gamma concentrations decreased and antipurine clearance increased. And decreases in metabolism for multiple substrates have also been reported after direct administration of interferon alpha to hepatitis and myeloma patients. However, again, I want to mention that other cytokines such as IL-6 are also increased in these diseases, and that changes in antipurine clearance in subjects with respiratory infection or HIV patients treated with zidovudin are generally more limited compared to the changes in rheumatoid arthritis patients given IL-6 receptor antagonists. Therefore, it is unclear if interferon alpha or interferon gamma are the main cause of the CYP suppression or not. For TNF alpha, in congestive heart failure patients, given a metabolic probe cocktail consisting different CYP substrates, a significant inverse relationship was found between TNF-alpha and CYP2C19 activities, but not for CYP1 and 2, 2D6, and 2E1. However, in those same patients, IL-6 was also significantly inversely correlated with CYP2C19 activity. So we don't really know if this suppression was due to TNF-alpha or IL-6. In another study, higher TNF-alpha concentrations in HIV patients were significantly correlated with reduced 3A4 activity, but not 2D6 activity. And also in ovarian cancer patients, TNF-alpha was associated with increased 2E1 activity and reduced 3A4 activity. However, again, the changes in enzyme activity were also significantly associated with IL-6. And another example, in psoriasis patients, TNF concentration also increased, but there's no correlation found between TNF-alpha levels and 2D6 activity. And in COVID-19 patients or surgery patients, they did not really find a correlation between TNF-alpha levels and the decreased CYP1A2, 2C19, and 3A4 activities, since these decreased subactivities could be due to IL-6. And green box here shows a study of therapeutic protein drug interactions using etanercept. Etanercept is a fusion protein that blocks TNF-alpha from binding to its receptor and therefore can remove the suppressive effect of raised TNF-alpha on CYPs. And in this study, administrating etanercept to healthy volunteers had no effect on Jackson warfarin exposure. However, the caveat of this study is that healthy volunteers would be expected to have low circulating TNF-alpha levels to begin with. So therefore, any potential suppression prior to etanercept in these subjects probably would be minimal. For other cytokines, in patients with psoriasis, IL-17 concentrations were significantly increased when compared with healthy subject, but there's no correlation found between raised IL-17 levels and venlafaxine, which is a 2D6 and PGP substrate. And bimacizumab, which is an anti-VEGF mAb, was administered to non-small cell lung cancers patients with erlotinib, and the exposure of both drugs were compared with that in patients receiving each drug alone. And there's no differences found in erlotinib PK when bimacizumab was co-administered together, suggesting that VEGF probably does not have a suppressive effect on CYP3A4. Okay. So here I'm showing some of the PBPK modeling examples in the literature, and data for the in vitro suppression of CYP activity or mRNA from that Dickman study I mentioned earlier have been used to successfully describe the clinical consequences of CYP suppression by IL-6 in PBPK models. The figure, the font is kind of small here because the figure is very big, but you can look at it in our paper. It's really a summary of observed and simulated effects of chronic and transient IL-6 elevation using PBPK modeling. And the black and blue bars represent predicted and observed data, and the white, white, yellow, pink, and red areas represent insignificant, weak, moderate, and strong interactions, respectively. And a few models are designed to capture the therapeutic protein drug interactions in patients who have chronically raised IL-6 levels, even at the highest IL-6 concentration tested, 100 picogram per ml. Moderate drug-drug interactions were predicted only with CYP3A4 substrates. And on the other hand, a few models focus on predicting transiently raised IL-6 levels, where IL-6 levels increase rapidly to 1600 picogram per mil after blinatumomab administration, or 50,000 picogram per mil in COVID-19 patients, for example. And out of the predicted effects of transiently raised IL-6 on small molecule exposure were also small, with all changes within twofold. So in conclusions, in part 1, we see that in vitro data suggests IL-6 is the most potent suppressor, and CYP3A4 is the most sensitive isoform. Interferon gamma and TNF alpha appear to have reduced or similar suppressive effect, although they may cause greater effect on CYP1A2. IL-2 and IL-1 beta could lead to minor CYP suppression. IL-10 does not cause CYP suppression, but data for other cytokines are lacking. And also incubation of cytokine cocktails with hepatocyte suggests similar extents of CYP suppression for combined cytokines compared to IL-6 alone. For in vivo data supporting a direct effect on CYPs are inconclusive for most of the cytokines, except for IL-6. And those reported interactions could be simply driven by IL-6. And although raised levels of IL-6, interferon gamma, TNF alpha could lead to CYP suppression, the cytokine levels observed in common immune disorders are generally lower than in vitro EC50 values, and therefore mostly we see weak or moderate drug-biologic interactions. However, transiently elevated concentrations after dosing of cytokine modulators may be close or above the EC50 values. And by incorporating in vitro mRNA EMIN EC50 values of IL-6, PBPK models are able to predict concentrations of CYP substrates in both chronically raised or transiently raised IL-6. And with that, I'll pass the stage to Xian.
Okay. Thank you, Freddie, for the informative presentation on in vitro and in vivo evidence of cytokine-mediated CYP suppression. So building on that discussion, I'm excited to share with you a sneak peek of what is coming later this year in Simcyp version 23. It's a new module to mechanistically model the interaction between therapeutic protein and a small molecule drug. So in the next few minutes, I will give you a bit of background and a preview of what you can expect from this new module, including the module structure, data collation, and case study demonstrate its potential. So in 2020, FDA released a draft guidance of DDI assessment for therapeutic protein. Specific mention the utility of PBPK model in providing mechanistic understanding of DDI potential for therapeutic protein. And in July last year, ICH released a draft guidance M12 on the drug interaction study. So both FDA and ICH suggested when evaluating therapeutic protein DDI potential, the sponsor should consider the DDI mechanism, clearance of the therapeutic protein, as well as the co-medication in patient population. So one example of therapeutic protein DDI is ADC small molecule payload interacting with other small molecule drugs. This can already be done using Simcyp simulator. And another example is this DBI with cytokine or cytokine modulator, which will become available in Simcyp version 23. There are several reviews and perspectives published from regulatory authorities, pharmaceutical industry, and also outside from Simcyp on this topic. Well, in terms of the PBPK models, they are mainly focusing on the IL-6 and its suppressive effects on CYP3A4. In fact, majority of them has used the Simcyp through a workaround approach, where IL-6 is modeled as a small molecule drug following continuous IV infusion to mimic the elevated IL-6 level in patient. And the prediction is fairly good, as Freddie has already shown you in that forest plot earlier. Then the aim of this project is to mechanistic model this DDI between therapeutic protein and a small molecule drug that related to outer level of cytokine, which regulates the expression of CYP enzyme. So here is a complex DDI network implemented within Simcyp simulator for the small molecule drug. And essentially, this project will bring the therapeutic protein to this DDI network through modulating CYP enzyme expression. So this new module will enable the user to model the cytokine or cytokine modulator as a large molecule, accounting for its DDI mechanism, clearance of therapeutic protein, as well as the co-medication in patient population. That is exactly ICH and FDA has suggested in their guidance. The mechanism of cytokine-mediated CYP modulation is apparently transcriptional, although the mechanism can be cytokine-specific. So this graph illustrates some plausible mechanism for some major cytokines, including transcriptional downregulation of nuclear receptor, alters the liver transcriptional factors, or directly affects through the NF-kappa B. So given the complexity and the still limited knowledge of the mechanism, so to model the cytokine-mediated CYP modulation, we are focusing on the crosstalk among the cytokine receptor, and the cytokine modulator. So our team had looked into the IL-6 case because this is one of the most interest. There are two known IL-6 signaling pathways called IL-6 classic signaling and trans signaling. So IL-6 classic signaling requires a membrane-bound IL-6 receptor and is restricted to hepatocytes, some epithelial cells, and some leukocytes. While the trans signaling requires a soluble IL-6 receptor, which is cleaved from the membrane-bound receptor and binds to the ubiquitous expressed GP130, and this can happen almost on every cells. These two signaling pathways tend to trigger different downstream events, as suggested in some reviews and articles. So our question can both classic and trans signaling lead to CYP modulation? And do we have sufficient data to differentiate two signaling pathways and model them separately? Well, the quick answer to the first one is yes to classic signaling, as suggested based on some literature, either direct or indirect evidence. And the answer to the second one is no. So therefore, our model will focusing on modeling the classic signaling pathway and account for the CYP modulation in liver and gut, where the membrane-bound IL-6 is expressed. And here is the schematic of proposed mechanism, looking at the three typical scenarios. So cytokine binds to the cytokine receptor and initiates the intracellular signaling, which leads to the transcriptional downregulation of some CYP enzyme. When the cytokine receptor antagonist or cytokine anti-cytokine antibody kicks in, it blocks the cytokine from binds to its receptor. Therefore, the suppressive effects of CYP enzyme will be removed and CYP activity will be restored. The suppression effects on CYP will be modeled using the same turnover model as we use for the small molecule drug, where the suppressor reduces the synthesis of CYP enzyme. And moving on to the next slide, I will take the cytokine receptor antagonist as an example to walk you through how we are going to link biologics model with small molecule model through the cytokine-mediated CYP modulation. So here we have the biologics model, small molecule model, and the one that describes the crosstalk between cytokine receptor, and the cytokine receptor antagonist. So the concentration time profile of cytokine receptor antagonist is modeled by biologics PBPK and fit into this crosstalk model here. The cytokine receptor antagonist blocks cytokine from binding to the receptor, reduces the level of the cytokine receptor complex, and subsequently relieves the suppression effects. Then the restored CYP enzyme lead to the increased metabolism of the small molecule drug, and as what you can see, the lower exposure of the small molecule drug. The alteration of cytokine levels and the clinical interactions have been observed in inflammation disease, in the cancer, or with the treatment of cytokine products or the cytokine modulators. Besides, there are also cases where cytokine levels is elevated transiently after therapeutic treatment. With Freddie already giving you quite a lot of details about that. But what I'm going to talk in the next few slides, I'll just show you a case of modeling the IL-6-mediated CYP suppression and DBI simulation using this new module. So we start with the data collation. So on top, you have see that this is the schematic showing the parameter and also what required to describe the IL-6 and IL-6 receptor interaction. So for IL-6 receptor is this symbol R here. We need to know its level, its expression, membrane-bound expression in liver and gut, and also its synthesis degradation rate. And the C symbol here represents the IL-6, where we can model that through the biologics PBPK model to get this level at different organ. And then the interaction between IL-6 and IL-6 receptor can be modeled either through the full model, the K on and the K off, or you have the assumption about the quincy equilibrium approximation, which is the KD what you will use in that model. Then for this complex, we need the internalization rate. So the team had looked through the literature carefully, and we were able to collect the data that for us to parameterize this model. Then next, we have built the IL-6 large molecule model using Simcyp version 22 to describe its PK profile. Then, as you can see on the bottom here, compared to the observed data in the literature where IL-6 is dosed intravenously, the simulated PK profile recovered observed data reasonably well. Moving on, we linked the Simcyp simulator to Simcyp Designer for prototyping. What I'm showing you here is to simulate IL-6 interaction with midazolam as an example. So the change of the midazolam exposure before and following the elevated IL-6 level can be recovered by this model, as you can see from this left bottom figure. And we're also showing you here a few different time courses. They are like 3A4 enzyme level change over time in liver and gut. Also, the IL-6 receptor complex formation in liver and gut over time. This is the driving force for the CYP3A4 suppression model. And before I wrap up, I'd like to show you a beta version of the user interface that we're currently working on. So you can see the firsthand how it will work. First of all, we will introduce additional compound slot called the TP modulator, where the user can load either cytokine or the cytokine receptor, cytokine modulator for DBI simulation. And on the list of the modules for the TP modulator, apart from those that you already seen for the large molecules, we are introducing an interaction screen for the user to incorporate the CYP suppression or induction parameters. And in order to model the DBI with cytokine modulator, cytokine and the cytokine receptor interaction will be defined on this TMI screen, which is target-mediated interaction model screen. And the cytokine modulator interaction with either cytokine or cytokine receptor can be described in the current function, using the current function in the TMDD screen. So, and we understand that this is the new approach, and they may require some training and support. Apart from giving you this new module, we will also be providing tutorial, workspaces, and other sources necessary to help our users become familiar with this new module and input the corresponding module parameter in the right places for handle different scenarios. To summarize my talk, really the aim of this project was set based on the ICH and FDA guidance. And what you can have is to link the large molecule model to the small molecule model to simulate the DBI related to the cytokine-mediated suppression or induction. We're going to model the crosstalk among the cytokine modulator, cytokine and cytokine receptor. You'll be able to handle complex DBI scenario with one small molecule substrate, one TP inhibitor, and up to 4 small molecule inhibitors. There will be user-friendly interface, and we're also going to provide the tutorial and a workspace to help the user become familiar with this new module. And of course, we are working on the case study. I already showing the example. Those will be used to evaluate the model performance. Although the model will not be in specific antibody and oligonucleotide for version 23, but the DBI with this type of therapeutic protein can still be assessed semi-mechanistically through modeling just to elevate the cytokine levels and the magnitude of CYP suppression. While this model is not yet available, I hope this talk will give you a taste of what is to come and the possibilities that this new module will bring to the drug development. We're excited to continue working on this development and looking forward to share more updates with you in the coming month. And here is the team. With that, I think we're done with the presentation itself, and we're going to open up the floor for the discussion and the questions. Thank you.
Thanks, Freddie, and thanks, Jean. And yeah, thanks also to Kate for joining us as well to go through any questions that we have. Please raise your hand, or yeah, someone's already done that, as I suppose. Do you want to speak?
Hello, I'm Emily from Janssen. So yeah, thanks for this very interesting presentation. I wanted to know if you are able to capture the variability that we can observe in the IL-6 kinetics profile, especially during the CRS syndrome, because for in like array population that's quite stable, but are you able to capture the high variability that we can see during CRS syndrome?
Sorry, is that with the new model or with the models that have been published so far?
No, with the new model.
Okay, with the new model, I haven't evaluated every different diseases and related variability, but that's definitely something we're going to look into and to see how they capture the variability and the such.
Okay, thanks.
Are there any more questions online? Yep. Jennifer?
I just had a question about the population aspects. If we are doing such a DDI, are you looking at, if you have already presented this, I'm sorry, I might have missed it, like whether we're looking at a cancer population or a different special population within that DDI database, where you have the biologics, the small molecule, and the substrate interaction kind of setup.
Okay, I guess it's also asked the new module. Yeah, so it's definitely something we're going to look into for sure. And you can definitely do that for different, because in Simcyp we already have a cancer population or different disease population, and those will be all available. This new module will be available for all of them. So you definitely can load them with that. And when we're doing that, the one thing you might need to pay attention is because at the moment, the suppression effects on 3A4 or maybe some other enzyme has already been incorporated in certain population. So you might need to reset that back to the healthy volunteer level and then use our model to suppress it and see how they're going to pick up the data.
Okay, makes sense. Thank you.
Has anyone else got any questions for the panel?
So are you suggesting to not use the array population which is currently available in Simcyp, or and now use this IL-6 module instead?
Yeah, I mean, so for the disease population, we already incorporate what you have in that disease population is all the physiological change and all other things be incorporated in. We suggest you use that, but there one change you might need to make is for the 3A4 abundance, because at the moment we already have a lower abundance that could be partly, or at least partly, due to the higher IL-6 level in certain disease population, and then you need to set up back. Otherwise, you're double counting the suppressant effects, and you will not get the data. The simulation right to recover the observed data. That's what I mean.
And one additional question. So, like in the Macavalam paper, you applied first inactivation or suppression on the liver, and then there is an extra step to apply this inactivation in the gut. With this IL-6 module, will it be in one? Can we do that in one go, or do we still have to do it in 2 steps?
Yeah, that's a very good question. Of course, you're gonna do it in one go. That's one of the purpose we want to do this whole model. You don't have to run 2 simulations and set up different things. So we're gonna account for the suppression. Well, if talking about 3A4, the suppression effects in both liver and the gut. So yeah.
Okay, thanks.
Sorry. I have a follow-up question to that is, if we want to use the cancer population as is, but then add the effect of IL-6 from like a drug-induced IL-6, could we use this module with the cancer population without adjusting the base parameters of the cancer population itself and just add the component of the drug-induced IL-6 to this using the biologics module, or does it have to be a total effect IL-6 overall?
I think maybe Kate can add to that. I think it depends on you're talking about the chronic effects or transit effects. What I can think, if it's transit effects after you give it the therapeutic protein, you see the transitly elevated IL-6 level. I think can just use the cancer patient as it is. But if it's a chronic condition you're looking at, I think you need to reset that to avoid the double counting of a suppression. That's what I think.
Yeah, I would agree with that.
Okay, so if it's like an appear-disappear effect
Yeah.
due to drug, then you can use it in association with the special population. Okay.
Yeah, I think so.
Thank you.
It can be something that we can take a look at once the models are available as well.
Yeah, this is Masood. I just wanted to add what Kate just mentioned. We will look at it because the baseline would be based on the value that you have in population. So we have to look to see whether we go with the like healthy volunteer as a baseline in all population, or if we found general as a baseline for cancer population, which will be a bit more challenging to do that. So this bit, it needs a bit of extra work.
Sure. Thanks, Masood.
Okay, I can't see any more questions coming in. Thanks again to Jean, Freddie, and to Kate for all the hard work putting this together, and obviously Jean's continual work to get it in for version 23, as well as the rest of the team. So thanks again. Do you mind just moving to the last slide? And what we'll do is just as we close, just to let you know about the next meeting that we're going to have on the 17th of May. And the publication that it relates to is a publication by MEMA, and looking at hepatic impairment population, relating to some of the updates that were made in version 21 as well. So that'll be a really interesting presentation. So that registration will open soon, so look out for that on the members area. For those of you who haven't logged in yet, there is a new members area anyway, so perhaps try and look out for that and do that in advance as well. Okay, so thanks again, and we'll speak to you next time. Thank you.